have all of you here. We'll start anyway. The last session of the summer school is really going right into the farm, shall we say. So you've heard about how you sample, you've heard about the various microbiome analyses, you've heard about microorganisms and DNA sequencing. Now we're going to move right forward to think about how we can use that to improve the production on the farm. We have two speakers in the last session who in some senses are going in opposing directions. Stefano started with breeder organisations and is now working very heavily in research and application, translation of application. Riccardo, on the other hand, is a researcher who has gone into the breeding organisations and is looking how to move research into the field. So we start with Stefano, who will give us a background on genetic and genomic selection as it was and how it will be, and then Riccardo will tell us a bit more about the visions of using genomics. Okay, thank you.
Thank you very much, John. Good morning, everybody. Yes, as John has told you, the topic of my presentation is something possibly not completely related to what you have seen so far, but I will try to convince you that what you have seen so far is actually the result. I'm sorry. What we have seen so far is actually the result of something that we did in the farm. It's something related to the choice that the farmers and the breeders does at farm level. Basically is the result of selection. So what we see in the animals is not only the results of the environment, but is the results of our selection choices. And so I would like to give you a quick overview of how the things worked when there was no genomic tools, how the things work now in the what I call the genomic era, and what we can do in this new situation that has been created by the new tools introduced in the field of genomics. Okay, so I think. Sorry, just let me. Okay. First of all, let's try to understand what we mean by genetic improvement. You know, it seems to be trivial in the sense that genetic improvement is trying to improve the genetic potential of our individuals. Why this is important? It is important because genetic is a part of the results that we're going to have. So genetics is with nutrition, with management, with sanity, all those things will be increasing our incomes. So it's a very important part. And it is even more important if you look at this equation, because this is the basic equation and everything is related to this. So this is the phenotype, so what we observe, anything that you observe is the phenotype. For instance, even rumen composition or milk production or everything you can see is the results of two components, basically, the genotype and the environment. And plus another component, a little bit more difficult, the interaction between the two. So genotype and environment. And the genotype are all the genes which regulates the expression of the trait. And this is what I was looking for and what we aim to improve. And the environment is everything that it's not genetic, so the farm, the weather, feeding, and so on. So, but what is the main important point? The main important point is that the genes can pass through generations, so if I select the good ones, I will be improving my breed. But
just like John.
and the most important point is that genes, if you select the right ones, you can pass through generations. On the contrary, environment can change over the years, so you don't have a total control of the environment. So what you can do is try to select the best that you can and then try to control the environment, but not completely. Well, this is the Scottishism. It's not so variable, but you can see we have the same genes more or less, but the weather is changed. Okay? But how to improve genetically our animals? So first of all, the first step is to identify what we call the breeding objective. You know, this seems to be trivial again, but you need to know what you are going to do with your animals. So what I would like to do, I would like to increase milk production. Okay, so my breeding objective would be a kilogram of milks, for instance, or the quality of the milks in terms of protein, for instance. I would like to improve the functionality of my cows, so I'm going to select for animal with good reproduction or with less mastitis. Mastitis are infection of the udder, which cause several problems, especially economical problems. Or, for instance, a very new and up-to-date breeding objective is reducing greenhouse emissions. Yes, I can do that if I select for feed efficiency, for instance. So this is the phenotype, and I would like to improve this phenotype. So this is the first step. The second step, another very important step, and my colleague just before me told you about this, data recording and data collection. It's extremely important to record correctly all the information. So another obvious information, the animal ID. Sometimes you don't have the animal ID, you mix the animal ID, so it will be a mess if you don't control regularly. And then you need to record the phenotype. So if you want to increase milk production, you have to record milk production. If you want to increase feed efficiency, you need something to record feed efficiency, and so on. So then what we have, another very important aspect, that is this one. So if I have a trait, I want to know if the trait is related to genetics. So the trait improvement is based on genes or not. So we do have to estimate some parameters which allows us to make selection. And one of these is what we call heritability. The heritability, this is the symbol, is a parameter which tells me how much of the variation that I see in the trait is due to genetic. So, for instance, just to give you some examples, some quick examples, if you look at milk production, we have an average value for heritability of 0.30. Heritability vary from 0 to 1. So 0, it means no effect of the genotype, and 1, total effect of the genotype. So you can understand that the higher the heritability, the best results in terms of selection. You can see that heritability depends on the trait. So you have milk production around 30, reproduction, very difficult trait, around 10, enteric methane emissions around 30 as well. So, and as I told you, it is extremely important. Sorry, the progress. Okay, so this is extremely important. If you have no genetic variation, you have no party. You cannot do selection if you don't have genetic variation. So it's extremely important, this point. So, okay, so we collect the data, we see if there is a gene impact on the data and so on. But then we need tools to select our animals. And generally speaking, the tools used in selection are called estimated breeding value. I have to be able to give a value to the genetic potential of the animal. So, and the terminology is EBV, that stands for estimated breeding value. And you can see here, an animal breeding value is the estimate of its genetic potential. How much genetics you can pass to the next generations. Okay, so and you can easily understand that the higher the value, the best. And generally speaking, for instance, these tools are expressed in the units of measurement. So if you are selecting for milk, you will have some value in the scale of milk. So a breeding value of a bull with a breeding value of 2,000 kilograms, it means that it will pass to its progeny 1,000 kilograms potential to make milk. The genes that can enable the son to make more milk, the daughter. And then you can have genetic or genomic breeding value, and I'm going to tell you what the difference is between these two. Okay? So I told you, fixing the breeding objective, what we are going to do, collect the data based on the breeding objective, look at the genetic aspects of the breeding objective. Is it genetically related? Is it related to genetics or not? And then try to develop some tools to select animals. You know, when you have to select something, you need a measurement, a parameter to select the animals. So the parameter in this case is the estimated breeding value. Okay, well, when you have all those things, you can, let's say, using the results and setting up a breeding plan. What I mean, you can select your animals, you can select the best bulls, you can select the best cows that you have in your herds, then you can mate them in order to obtain a better animal. Okay? And so you have a so-called breeding plan, you select and mate, and you expect to have genetic progress. Please take in mind that at least for dairy cows, to do all these things, it takes several years. Approximately 5-6 year of generation interval. If you do something now in terms of genetics, you will see the results in 5-6 years, a generation approximately. Okay, and this happened with no genomics. Well, and the objective is obviously to get genetic progress. This trend is just the milk production in terms of the genetic trend for milk production for the Italian Holstein. So you can see that this is the year of birth. You can see that in over 10 years the increase due to genetic selection is actually very, very high. Okay. But please do remember another thing, that do remember the equation that I showed you before. The equation, the phenotype is genotype plus environment. So you can have very good animals genetically, but if the environment is not good, you will have no results or even negative results. Okay? But what happened with what I call the genomic revolution, with new information has arrived, new data, new tools. So what happened apart from the terminology? So we have introduced new names like SNPs, genomic EBV, or something like that. Apart from the terminology, there are some other important things like the reduction of the generation interval. It means the time between the birth of a bull and the birth of his daughters. So if you can reduce the generation interval, you will have a faster genetic improvement. Okay. Another important thing, I told you about EBVs, the estimated breeding value. The estimated breeding value is an estimate. So, as any kind of estimates, it has an accuracy. And the accuracy depends on the number of data that you have and the type of data that you have. So, one of the points of the genomic revolution is that we are going to have more accurate estimated breeding value, and this is extremely important. If you use the breeding value to make a choice, and the breeding value is not accurate, you are not going to get good results. Okay. So, but if you are using accurate tool, you are going to get good results. So, this is extremely important. And then another aspect is related to the possibility to analyze new traits, more complex traits, and this is another aspect of the genomic revolution. But why we talk about genomic, what does genomic mean? Well, basically you can see here, genomic is the study of the genome, the living organism, in the sense that we are interested in the structure, function of the genes, the evolution, all these kind of things. But for sure, what happened over the last year is that new methodology like genotyping and sequencing, a lot of increase in technology, several international projects have boosted the use of the genomic information. So what was very difficult 10 years ago is now very easy to genotype an animal, to look at the genome of the animal is quite easy and is very less expensive than before. And then we do have, another aspect is bioinformatic tools, and that we didn't have in the past. You now have millions of records for each animal, and you have to have enough space to store the data. You need to have the right computer to analyze the data. You need the right algorithm to analyze the data. So bioinformatic tools is another very important aspect. But what happened before, what I call the genomic revolution? I told you about the breeding value estimation. How did I do the breeding value estimation? I basically used two types of information. The phenotype that I collected in the earth, so milk production for instance, and another information is simply the pedigree of the animal. So I went to the farm, I collect milk production, at the same time I collect information of the father of the cow and the mother of the cow, and so on. So this is called pedigree information. And what I did then, if you want to evaluate a bull for milk production is a little bit difficult because bull doesn't produce milk, but his daughters produce milk. So what you can do is to evaluate the genetic potential of a bull through his daughters. This is progeny testing. So estimating the genetic value of an individual using the performance of his or her offsprings. And I think that you can imagine that if this takes a lot of time, because you need to wait for the daughters of the bull start producing milk. So you need to wait at least 5 years, or 4 or 5 years, before a bull can be evaluated. You want to estimate the genetic potential of a bull? Okay, that's okay. The bull born today, then when it is 1 year old, you can start to inseminate the cows. You need to wait for the cows to calve, and you need to wait for the production of the daughters of the bull. So it takes a gestation is approximately, like human is 280 days. So you need more or less 4 years before you have the data to evaluate the bull. So a lot of time. You need to wait 4 years. And then when you have the data, you can evaluate the bull and start using as bull of other bulls and so on. So the process is quite complex, and especially, apart from being complex, is very long. It takes a lot of time and a lot of money as well, because you have to collect a lot of data, wait for the data to be collected and so on. So, and then there is another thing that I always like to tell about, is a sort of paradox, in the sense that we are estimating the genetic value of an individual without look at the genes, actually. I'm just saying that the genetic value is 2000 or 1000, based on what? Based on production and based on relationship. But relationship just father, son, and just this, not based on what it is actually in the genome, okay? And this is a sort of paradox. But this was not possible because there wasn't the information to do that. Okay? But now there is. This is the very the very big difference. So what happens now is that we can, well, first of all, read the complete genome of an animal, and apart from this, we can discover some particular area of the genome where there are some variation, what we call SNP, single nucleotide polymorphism. You can see here. So I can discover areas of variation, and I can use this information to estimate a more accurate breeding value. And I can do that even because now I have some additional tools that allows me to analyze a lot of data, a lot of animals with very few money. So those are some SNP chips that I can use to genotype all the animals, so to discover the SNPs that I was telling you about. Okay. So what happened now is that I have a lot of animals and I have this lot of information. Those are all the variation area that I was telling you about. So you can see here I have a lot of information, and I can use this information to estimate the genetic value. But obviously, how can I do that? So if before I just used the phenotype and the pedigree, now what I can do is to add some other information that is the genomic. So all this new information that I have been collecting. Okay, but how can I estimate the value of each of these points? This is the most difficult part, but let me give you a very small example. Okay, let's suppose that I'm going to buy fruits in a market. Okay, so I'm going to visit 4 different markets. Okay, and I'm going to buy the same type of fruits but different quantities. And you have here, so in the first market I buy 5 pears, 2 apples, 2 peaches and 3 kiwi, and I spend 23 euros. Then I go in another market and you see there's different quantities, same fruits, and so on and so on. Now, probably for you this means nothing, but for people like me it means something. This is a set of equations, and with this set of equations, those are the variables that I can estimate. So I can solve the set of equations and I can estimate the value, in the arrow value of 1 pear, 1 apple, 1 peach, 1 kiwi. So what? Well, with this information, those estimates can be a market expected cost. I can estimate how much I'm going to spend in another market without seeing the market, because I estimated this based on previous data. So, and the more markets we visit, the more accurate these estimates will be. If you move back to cattle, we have a lot of animals. There's SNPs. I'll show you. I have, for some animals, their traditional estimated breeding value. So I can use those previous information to estimate the value of the SNPs, and I can use these estimates in a completely different animal. So I can estimate the value of any SNP, we can genotype an individual and estimate the value of the genomic value of the individual. Obviously, you can, I think that you can easily understand that everything depends on how well I'm going to estimate the SNP value. So if I have a lot of information here, if I visit a lot of markets, I'm able to develop a good estimation of how much I'm going to spend in a different market. Okay, so this is really impressive difference between the traditional system and the new system. It's extremely important because you can, for instance, you can estimate the value, the genetic value of an animal when he is born, or even at embryo level, because what you need is the genotype of the animal. So you can understand that the time of selection is, the generational interval is extremely reduced. Yes, sorry.
But still you say that you can estimate the value at the embryo level, but again you have the environment and the environment in the mother during the pregnancy. It probably also affects the genes, right? The gene.
Well, this is a good point. It could be a good point, but at the moment, as far as I know, nobody has looked at... I mean, there are some works about the effect of the mother on the genotype of the son. But I think that in this case, it's not really important, even because what you are going to do is using estimate of the SNPs based on previous information estimated on other animals. So, yes, but it's a very good point. I think that I was thinking about. I think that in some cases you can control the development of the embryo at the lab. So the effect that you are telling me it's not so important, I think, but I'm not so sure. Anyway, it could be a possible source of variation, but I can't tell you how much is the impact of the mother. If I remember correctly, in some previous studies, the impact of the mother was around, on traditional evaluation, it was 2 or 3% of the genetic variation. So it was quite small. But it could be a good point, yes.
I'll make a comment after.
Yeah, okay, yeah. Very important is with this new methodology, what we are going to get is to increase the accuracy of the estimation. You can understand that you make a better choice than before. And basically you reduce generation interval because, yes, you can select animals at birth and you don't need to wait until the daughters will be born.
Sorry, but you use genomic information to estimate the breeding value only, or you are going to use it as additional information with pedigrees and phenotype? Because if you use this, you will reduce generation interval. But if you are going to use it as additional information, so in the end you need to the pedigree as well.
Another good question. Let's say that at the beginning, this methodology started approximately 4 years ago, more or less. So 4 years ago, you didn't have a lot of estimates available to develop a very accurate methodology. So what you did was to use both information, the traditional and the new one, and you use some statistical approximation to join the two informations. At the moment, what has happened is that you have a lot of animals with the genotypes. So the weight of the pedigree, of the traditional pedigree, has decreased a lot. So when you have an animal, so the weight of the pedigree is basically very, very, very reduced. So you don't need any more such an information. That was how the system started some years ago, 4 years ago, but now it's basically based on, only on genomic value. Okay, let's say that it's like you have very few markets and you put in the markets the effect of the town. But now you have so many markets that you don't need the effect of the town anymore, where the markets are located. Okay. So, but this was just to give you the idea that the two information, at least at the beginning, were come together for a while, and then I'm using essentially genomics. The other important aspects are the increase of selection intensity and the increase in rate of genetic gain. If I can select better my animals, I for sure will get higher genetic gain, and at the same time I have the possibility to increase the selection intensity in the sense that I have the possibility to look at all the animals. In the sense that, think about it, in the past when I used the progeny testing system, I had to select the bulls which will be entering the progeny testing. So very few bulls because it was expensive. Now I can look through all the population, so the probability to get a very good one has increased because the sample that you are using is larger than before. But this is just one of the things that we can do with genomics, with the information of the genome. There are some other things that we can do. For instance, we can test the parentage of the animals, and another very important point in cattle, and we can do this using SNPs, using the variation at genomic level in place of microsatellites. And this is something developed a couple of years ago, and you know, from the international organization, the International Society for Animal Genetics, and you can use SNPs in place of microsatellites to verify parentage. This is another very important step forward. And then another aspect, you can use genomic information to look at recessive defects. For instance, another very important point in dairy cattle, and the results of selection has brought a lot of problems in terms of recessive defects. And what you can do with genomics is having the possibility to detect or find the region, the exact region of the genome where something happens. So this is just a very, this is just an example. I had thousands of now, there are thousands of papers like this, and this is quite recent, and it's quite interesting because it talks about fertility, a very important aspect. But at the same time, you can do something more, even more complex traits, like for instance paratuberculosis, bovine paratuberculosis, a very important disease, and genomics gives you the possibility to look at the genetic aspects of such a disease. Or if you want to look at something different, for instance feed efficiency. Feed efficiency is another thing that they are looking at using genomics, to see if there is some relationship between feed efficiency and the genes. But now, I think that I told you about genomics, more or less, what you can do, what you cannot do. But a very important point, this is the first one, and yes, we have to come down to earth in the sense that if you don't have the right phenotypes, you are going to do nothing with genomics. So you need the phenotype. The big difference with what happened before is that you can collect probably less phenotypes, but you have to collect more accurately. So if you have an accurate phenotype, you can use it with genomics to estimate the breeding value, to look for causal mutation or causal mutations, and so on. Then I think that you got the idea, I hope so, that using genomics you can bridge the gap, in the sense that you can get to results in a shorter time, very short than before, shorter than before. So let's say that you want to select for feed efficiency, probably now you can do, because you can collect less data and you can use it in a more informative way. And then obviously, if you look at the genes, you have, I didn't tell you about the relationship between the traits, another very important point, and possibly a lecture only for this, for relationship between traits. So with genomics, you should have the possibility to understand the relationship between the traits. So if you select for milk, at the same time you select for something else that you can't see, but you are selecting for. So this is very important. The last two aspects are that now you have a lot of data. So this is a problem that you should consider in terms of storing and in terms of analysis. So you need the right algorithm to do that, and so think about it. And the very last is that down to earth number 2 is that anyway, for instance, when you talk to farmers and you talk of genomics, of genetics, they always look you quite strangely, like you look at me now. But so it is important that then you are able to extrapolate real application from what you do. So, for instance, estimating a breeding value is a real application. A mating program is a real application. So that's all, I think, yes.